Okay, good morning to all of you. First of all, thanks a lot for being here. It's our pleasure to talk about our nucleic acid extraction systems. Special thanks for the organization for inviting us to be here to talk about between 20 to 30 minutes about our systems. Okay, I am Jayme Nunes. I work for Loccus Biotecnologia. Loccus is a Brazilian company focused on molecular biology products. That's the idea. We are at the market for 22 years and working very hardly on our Brazilian market. And a few months ago, we started to export our products to Latin America. So I personally have been managing this exportation. So it's a Brazilian company nowadays. We focus on Latin America, and for sure we are going to be in your country as well in short term. That's the idea. Let's talk about these tools. We are going to focus on nucleic acid extraction automation solutions. That's the idea. Let's start from the beginning. How can we do that? We are going to use magnetic beads. So the idea is to lysate the cell and release nucleic acid. That's the idea. By using proteinase K, by using lysate buffer. And after that, we have this kind of magnetic beads which associate with nucleic acids, even because this kind of beads, they have positive charges. That's the idea. So by association, we are going to have magnetic beads and nucleic acids association. After it, we can wash it by magnetic events and take off the proteins, membranes, and all that stuff. We are going to have the nucleic acids in an elution buffer. It can be water or tea. We can do it manually by using these kind of supports, magnetic bars, supports. We can use this kind of bars in tubes, small tubes, big tubes, or even plates. We can use it. We can have plates. In order to have it manually, it's so hard. It's a labor. It's a hard job. We can automate it. That's the idea. The automation by using magnetic beads. We can do it by extractor. But I mean, how can we make it available? How can we do that? By using the extractors. We have two ways to automate nucleic acid extraction. We can use liquid handler. I know that I think that you have seen it, liquid handler. But I mean, we can use solid handler. We can use this kind of particle movers. That's the solution I want to talk with you. Solid handling. That's the idea. We see the small extractor, extractor 16. And we see the big one. The big one is the extractor 96. We have another one, the third one, that's not in this picture. But I mean, extractor 16 is the one that we can see in front of you, personally. Let's talk about the system. First of all, how does it work? Look at that. I mean, you have the sample. You lysate the sample, you lysate the cells, you release the DNA and RNA from the cells. It's in the solution. After it, we're going to add beads, magnetic beads. Remember that magnetic beads, they have a positive charge. It will attach into the DNA RNA, which is the negative charge, by charge itself, okay? After it, you can wash, because the magnetic beads, It's with the DNA attached with it. We are going to put into the bar, magnetic bar. It will push, it will take the magnetic beads with the DNA or RNA. So we will be able to wash it, successive washers, 2, 3 wash. And after it, we are going to release the DNA from the magnetic beads by using these magnetic bars. That's it. That's the way. There is no tips. There's no liquid handling. It's a kind of solid handling because we are going to move particles one each other. That's the idea. No more than that. Let's go deeply and see how does it work. That's a Structa family. That's our Structa family. It's a Brazilian. And for sure, I mean, parts of the machine is being imported. But I mean, the assembling is in Brazilian. It's a kind of national product. It's a Structa 16. That's the small one. That's the smaller one. That's the Structa 32. It means that the first one, for example, they will be able to process from 1 to 16 samples simultaneously at the same time. The other one is the Structa 32, from 1 to 32 samples at the same time. That's the Structa 96, 96 samples simultaneously. That's a station. It's another solution. I mean, the idea is to start from the sample, and we are going to take the PCR plate and all the components that we are going to have in our PCR is in the plate. For example, the DNA, the isolated DNA or RNA, as well as TAC DNA, the NTP, buffer, magnesium, primers, and probe. It's all in there. You take the plate and you put it into the thermocycler. That's the idea. Okay? That's the biggest one. That's the biggest one. But I mean, what I'd like to focus on this talk is the Structa 16, which is the one that is in front of you. Let's talk about that. Steps: lysis, magnetic event, washing, and elution. All 4 events in the equipment, in the instrument. How can we do that? That's the kit. In our illustration is the kit. Actually, in one kit, we are going to have 6 plates. Let's take one plate and see it. In the plate, we are going to have the filled plate. We are going to have the buffers in there. So line 1 is lyse buffer. Line 2 is magnetic beads. Line 3, 4, and 5, washing buffer. Line 6, elution buffer. That's the first part of the plate. The second part of the plate is the same. Lysis buffer, magnetic beads, washing buffer, and elution buffer. We are going to take the samples and put it into the line 1 and line 7, which is the lysis buffer. It's going to release the DNA. You put it into the extractor. What does it have? What does it work? What does it do? They will take the magnetic bars, put into the line 2, attach the magnetic beads, go to the line 1, which is the released DNA and RNA, because it's the lyse buffer. Take the DNA RNA by magnetic event. Go to line 3, 4, and 5, washing steps. And go to the line 6, which is the elution buffer. Release the DNA RNA. Release the purified sample, purified material, genetic material. That's the sample. That's the purified material in 35, 40 minutes. That's the idea. Did you get it? So in the plate that you are going to have in the kit, it's a filled plate. It's a pre-filled plates. You have buffers, beads in the plates. It's ready to use. It's ready to use. Just you want to put into the sample, your sample into the line 1 and line 7, go to the extractor, put in there, and get it. And go to have your coffee. That's the idea. That's the kit for the Stracta 16, Stracta 32. When you get this kit and when you open the plate, you have to have 16 sample to be treated and get the purified material. Our solution, there's another solution for in the case you don't have 16 sample at the same time. That's the kind of having, for example, just one sample to be treated, just one sample to extract the DNA or RNA. We have this kind of strips, individual strips. So you take the strip like this, you just take the strip off, put into the support like this. That's the blue support. It's a kind of plate. You put the strip into the plate and go to the extractor, and you have isolation, extraction, and isolation in just one sample if you want to. So we are going to automate just one sample. That's the idea. On the other hand, you have a high routine, you have the Stracta 9.6. In this case, in the kit, you have 6 plates, but in one plate, you have 9.6 wells with beads, another plate with 9.6 well, elution buffer, washing buffers in other plates. So the machine is a kind of a carousel. It's going to move plates, and we will be able to extract the 9.6 samples simultaneously. That's the idea. That's the list of available kits. For sure, the one that you should look at is the pathogens. That's the pathogen. On the other hand, I mean, we have this kind of several kits for available for use. I mean, but the one that we will look at is the pathogen. In Brazil, we have Anvisa. Anvisa is a kind of regulatory agency, and several ones. It's available from Anvisa as well. It's approved by Anvisa for IVD proposals. Okay. We have for virus, DNA and RNA from virus, from blood is another one that is being validated. But I mean, again, the one that we should look at is the pathogen. Benefits, it's for sure. I mean, the benefits are speed, yield, purity, reproducibility, and you can scale up. You can start from Stracta 16, go to 32, go to 96 if you want to. It's easily scalable up. Let's talk about the quality control. I mean, as soon as you purify your DNA or RNA, you want to quantify, you want to qualify it. We can do it by fluorimetry. We can do it by spectrometry. You have both. You have just the fluorimetry and the spectrometry as well. We have this kind of tools available for you if you want to. We can check quantification. We can check the purity by ratios. Integrity we can check it by agarose gel. I mean, that's it. Let's go to the workflow. What do we have available as well in case you need it is a thermocycler. You have it in Brazil. I mean, it's being assembled in Brazil as well. It's AVD Anvisa approval. It's our Amplio 96. It's a 6-channel machine. Very functional. Very nice for the laboratory. If we're looking at the PCR machine as well, it's available, and it's a IVD this approval. And what we have worked now, and I mean we have launched it as well, is the master mix. We have been working hardly on this list of master mix. 3 of them is for DNA, 2 of them is for RNA. It's one step. In our P&D, they have been hardly working on trying to lyophilizate it and release it and give it to you as a lyophilized balls, dry balls, in order to reconstitute it in your laboratory, put it your primer, your probe, and go to the PCR machine, go to the qPCR assays. That's our challenger right now. But I mean, in a liquid form, it's available nowadays. I think that's it. I mean, thank you a lot for your time. I will be really more than happy to answer your questions. And please go to your to the extractor machine here and take it. Have your view on it. Okay, and I'll be more than happy to answer your questions. Thank you.
Vamos sortear isso daí. So we decided to invite those guys to present to you because one of the things we have to discuss as a consortium is how do we standardize extractions, right? In reduce user variability to have more quality samples in the biorepository. So they have a variety of options, but it's just to bring that up and discuss the need to standardize and automate processes, right? In all these countries, right?
Hello. Hi, good morning. I'm asking, I will be asking about presence of contaminating DNA from, for example, bacterial or other pathogen contamination. For example, if you'll be using saliva or other samples that might have pathogens that may contaminate the samples, would the DNA of these contaminating pathogens be excluded from the extraction process?
It's a nice question. Thanks a lot for that. Remember that, I mean, we are going to use the magnetic beads. So magnetic bead is a positive charge. They are going to attach into the negative charge nucleic acids. So it's general. The specificity will be in your next step. I mean, it will be in your PCR assays, not in the extraction. The extraction, we are going to have nucleic acids in general.
Okay, thank you.
Okay.